From MicrobeTV, this is TWiN, This Week in Neuroscience, episode number 70, recorded on March 9th, 2026. I'm Vincent Racaniello, and you're listening to the podcast about quantum relativity. The nervous system.
Things we know less about than quantum mechanics.
Oh, that's a good question. Well, let me introduce you, and then we can ask that. Joining me today from New Orleans, Vivian Morrison.
Hey, guys.
And from New York, Tim Chung.
Hello. Hi, Vincent. Hi, Vivian.
So Vivian,
Hey,
you think we know less about the nervous system than quantum relativity? Or vice versa?
I mean, I have heard physicists say that because we were talking about pre-show. Vincent and I are, just have been super into physics recently, understanding the nature of time and, all of that. And I have listened in these books to physicists say, like, yeah, it's just real hard to put together what's happening in the brain. I think because I don't know. They don't really say why, but maybe it's that we're not really sure what the equations are that we should be using to describe what's happening in the brain. Yeah, that's...
Well, I mean, the communication in the nervous system is by photons, right? Is that correct?
By
electrochemical
currents,
signals.
perhaps. Not necessarily photons, not as in like radiation. I think.
Okay.
But unless
so
we're
electricity,
we're starting to do optogenetics, in which case photons.
Yeah, right.
Oh, we're not talking about that here.
Electricity is the flow of electrons. Okay. I'm reading Feynman's lectures.
Oh yeah, those are
in
good.
Physics, and he starts off by trying to introduce you to everything. The chemistry, biology, and even neuroscience. Let's talk about how the nervous system works. And it's just so rudimentary. It's really funny.
Yeah.
But
Little molecules, little bubbles like bumping into each other.
very funny. But I like learning things that I haven't thought about, right? And current science communicators make it accessible. So I find Feynman and Neil deGrasse Tyson and others do a good job of explaining it. I can look at a 10-minute video just before I fall asleep and learn about space-time.
And then as you sleep, you'll consolidate that information. But we don't know how that happened. Well, we know some of it.
Okay, today we have by the way, folks, if you enjoy these programs, we'd love your support to produce them. You can go to microbe.tv/contribute. TWiN was the last podcast that I used to edit, and I recently handed it off to an editor because after recording, I would just forget about it, and it would be weeks before I got to it. Oh my gosh, I can't do this. So
and that's why people are like, God, why is the editing so bad on these videos? Or non-existent.
But now my editing skills will degrade because I'm not using them anymore, right? Because that's how things are. If you don't use skills, you will lose them. It's definitely true. But anyway, that's life. And if you'd like to support us, microbe.tv/contribute. And today, Vivian's turn to present a paper which hopefully will be more accessible than Hawking's book.
Oh, 100%. Yeah. I could say that I wanted to choose this paper because my brain is just not functioning too well after this 1 hour time change. Like, how does that always throw us for a loop? But I picked this paper a few weeks ago. So, and it's actually one that I read back when I was at Vanderbilt in 2019 when it was written. And it's a pretty straightforward paper, and that's a compliment. And we didn't. You don't have to work hard to understand cool things sometimes. And I've been kind of on a kick recently where I'm like, oh, brain waves and stuff, and that's fun because like you've said, Vincent, it's fun to learn about things that you don't know anything about, right?
Yeah.
But so this paper is just some like good old-fashioned electron microscopy, some immunofluorescent imaging, some knockout animals, and counting things. And the takeaway is still pretty cool. So I'm hoping this will kind of be like a light and breezy episode. So the paper was published in Nature Communications in 2019 by Kelly Monk's group. And there are two other names here at the end that are pretty famous in the glia field. That's David Lyons and Jonah Chan.
Hmm.
And the title is Myelinating Schwann cells ensheath multiple axons in the absence of E3 ligase component FBXW7, which I'll be calling FBOXW7. So right off the bat, we're going to be returning to something that I talked about when I first came onto the show, which are glial cells, and even more specifically, myelinating glial cells. And we've talked about myelin before, and I actually think a lot of listeners will know what it is because, we know, like, I think pretty much everybody knows somebody who suffers from multiple sclerosis, which is an autoimmune disease where the immune system targets myelin and the cells that create it. And it's like the most common cause of neurological disability, I think. So people know what myelin is, generally speaking, but basically it's a lipid-rich, fat-rich coating on the axon, which is the structure of the neuron that allows the electrical signal to go from, one place to its destination, from the cell body.
Like a covering on my wire, right?
Yeah. Oh, it's exactly like that. I mean, yeah, that's what people are always like. It's like an electrical wire, being insulated, so that the electrical signal can like get from point A to point B, and it can do it quickly and with high fidelity. And yeah, and I should say that the first author of this paper is Brianne Hardy. So well done, Dr. Hardy. I'm sure. I don't know where she is now, but so myelin is these concentric layers of cell membranes, mostly lipids, some proteins. It wraps around axons. And like I said, it helps the electrical signals travel more quickly and with higher fidelity from to their destination, which is the presynaptic terminal. And not all axons are myelinated, and not even in the central nervous system, and even in the peripheral nervous system.
Tim just left.
Yeah. Should I keep going?
Let's see if he comes back,
Okay.
because it's always a problem when people leave.
Mm-hmm.
So like everything else, there are myelination diseases, right?
Mm-hmm. Yeah.
Because the body goes wrong in every way possible.
Every way possible. Yep. Just can't escape it. Some people are born hypomyelinated, which is a big problem. Some people lose it.
But you said that some are not myelinated at all. But how do they get insulated then if it's needed for insulation?
Well, not everybody needs it. So for example, in the peripheral nervous system, a nerve is made up of a bunch of different axons of different types. And you can have a neuron that's mixed, or a nerve that is mixed. So it has sensory fibers that are taking information from the outside world to the brain. And then you can have also motor fibers that are taking motor commands from the brain out to the periphery. And so, for example, in axons that are for nociceptive or pain sensing cells, some of those are not myelinated. So the information travels more slowly, which is why you might have the experience of like you kick a table and then you're like, I kicked that table, or like I'm going to move my foot away from it, and then you're like, oh my god, why is this so painful, and why did it happen like 3 seconds after I kicked the table? So that's what I mean by,
It's quite funny that
for whatever reason,
evolution doesn't
Go ahead,
really,
Tim.
we've evolved to like have the slowest communication for the pain. So like evolution literally doesn't care about your feeling. It's the last thing. It's the last thing that
but
needs
maybe
to
that's
arrive.
what you need. You don't want acute pain to be the thing that stops you from escaping.
So you think that's
how I might think about it.
I thought
maybe
Because when you burn yourself on a stove, you pull away right away, and then the pain starts, right?
Yeah.
So the reflex happens right away.
And that's probably
like how many and
that might be because
Go
of the
ahead, Tim.
reflex arc inside the spinal cord that causes
Which
it.
doesn't
So
have
before
to
you're
go-
conscious of it, but even then, the pain going from your finger to your spinal cord is still relatively slow because they're not myelinated, and the evolution spent myelination on something else, which is Vivian probably talked about this already because I dropped out for a while due to poor internet.
Maybe not.
But it was for like things like proprioception, like how long, where your arms is, like, and how much your muscle tension is stretching. That is apparently much more important than pain, and that's why they spent the myelin on that. At least that's what I heard.
I think also the the reactions, the behaviors that would be initiated by acute pain are probably much simpler than the ones that the organism would need to take in the context of more chronic inflammatory pain, which is like, you got to find somewhere safe to be. You got to make sure that you got enough, you got to like shut down eating and digestion and whatever to funnel resources to, it's maybe like in a way, the more chronic pain signal is going to be more important. And at that point, it's like the danger is no longer there. So it doesn't need, anyway, that's all conjecture. Because I'm not really sure if we can know, but yeah, Vincent and I were talking about how not everything is myelinated. And so we were just saying that, certain fibers in the peripheral nervous system are not myelinated while others are. And those axons might exist in the same nerve bundle. So there's got to be some way to determine which ones get myelinated and which ones don't, which we'll get to. And then in the central...
Sorry, quick question.
Go ahead, Tim.
Do you know, since you know more about myelin than definitely me, do you know why, even though pain fibers aren't myelinated, when you lose Schwann cell myelin in terms of like Guillain-Barré syndrome, you do get pain, right? Like how does, even though the thing that is lost is like your proprioceptor fibers, you would think. Does anyone know why? Do you know why? It's all right. It's just wondering.
I don't know enough about that Guillain-Barré syndrome, and I was getting to this distinction, but since you brought up the name Schwann cell is the peripheral nervous system myelinating cell, and we contrast that against the central nervous system myelinating cell, which is the oligodendrocyte.
Actually, you know what? Now I
mean, the pain...
I think maybe as we go into the paper, we might know a bit more, because if I remember from this paper, I didn't know it until I read this paper, is that the Schwann cells still ensheath and wrap themselves around all kinds of bundles, including the small diameter pain bundles. They just don't form myelin.
Yeah. And
so when Schwann cells are in trouble, everything is in trouble.
Oh yeah, pretty much. And there's kind of I wanted to get back to just this question of what's myelinated and what's not. And I wanted to say that in the central nervous system, so the brain, particularly in the brain, because in the spinal cord, you'll see that there are huge like chunks of it that are they're white, and the white comes from the myelin. So like those areas are heavily myelinated, and those are fibers that are going, long distances up and down. And then, but in the brain, it's this kind of like messy, complex web of things. And researchers have found that an axon can be myelinated, but only in certain places. Inconsistently, the size of each individual myelinated section changes. And we're not really sure what the rhyme or reason is there. But it suggests that there's some sort of functional role for myelin that pertains to the timing of the arrival of action potentials at their destination, and the action potentials being that electrical signals that we were talking about, and that this what they call spike time or spike dependent time plasticity, or the things based on the arrival of those action potentials, is involved in regulating how circuits work and learning and memory, stuff like that. And these patterns, at least in the cortex, these patterns of myelination can be changed by learning a new skill. So there's like, I probably brought this up in a very, long ago or at some other point, that there are these really cool studies that show that these people who, they take a brain scan before they start the whole experiment, and then they teach these people how to juggle over like a period of 3 weeks, and then they do another brain scan, and there are these indirect ways of judging the amount of myelin and like which regions of the brain are myelinated, and they actually show that myelin in the motor cortex increases after this period of learning how to juggle.
Hmm.
So that's like obviously, well, there's got to be some connection there, right?
What about
Between increase in myelination and learning.
If you memorize a play, will myelin happen somewhere else in your brain?
In your like Shakespearean region?
Yes.
I doubt it.
I guess in the memory region, right? What part is for memory?
Well, we've talked about this before. There is no place.
It depends on. It'll be in the temporal lobe somewhere.
Mm-hmm. But I'm not
I can't
really
remember.
It's I don't know. I think it's complicated. But we're not here to talk about complicated things today. We're here to
the
talk
paper is
about
straightforward, right?
Yeah, straightforward.
Okay.
But I also want to bring up that we've been talking about myelin as a insulator helping to conduct this information, but there are also myelin is also known as a source of like energy resources, like metabolic support for neurons. So if you lose myelin, like you do in multiple sclerosis, you are gonna you might lose sensation for whatever region of the body is affected. You would lose the ability to like really use it, send motor commands to it. And if this happens repeatedly, such as in like relapsing, remitting multiple sclerosis, eventually the neuron, which is dependent on the oligodendrocyte to some extent for metabolic support, will become like weaker and weaker and can be more susceptible to neurodegeneration, which is one reason why these demyelinating diseases, they come and go, because remyelination is kind of a spontaneous process, but it doesn't happen super well. It gets worse with age. And then you've also got the health of the neuron then being undermined because of the loss of this metabolic support and ongoing inflammation. So beyond just this like electrical support, they also are metabolic support for neurons. So another thing that is a distinction between the peripheral nervous system and central nervous system myelinating cells that I think is relevant and relevant to the disease we were just talking about, is that the production of precursor cells and immature myelinating oligodendrocytes seems to happen pretty well, even in the adult nervous system following demyelination. But then it's that transition from an immature cell to a myelinating cell that kind of gets gunked up and gets stalled. And Schwann cells, on the other hand, are very plastic. So during nerve injury, for example, you can have a myelinating Schwann cell that goes, oh, it's time to, put on my different hat. It'll, detach itself or get rid of the myelin and instead take on this morphologically complex shape and take on a whole different set of tasks that involve like managing the situation. Saying, oh, yes, macrophages or immune cells, come here. This is where I want you to be. And I'm going to make a tube. And this tube is going to guide the neurons as they sprout and try to find their target area again.
Oh, they do that.
So if the Schwann,
do they do that during development, during neuroaxon?
That's a really interesting question. So I am not familiar with if the literature, if it exists, but this is one of the questions I had when I worked when I was at Vanderbilt, and I worked in a lab that studied Schwann cells, but I was like the central nervous system person, so I focused on oligodendrocytes. But I was thinking, So which comes first? Do the Schwann cells migrate out?
Because I heard
to like
myelination.
how do they know where to get? Is it like the axon gets laid down first because this the neuron, as it's like extending its little process, it's responding to all these chemotactic signals like go here, oh no, don't go there. Oh, I've arrived at my destination. And then do the Schwann cells migrate along that and then set up shop? Or are the Schwann cells also aware of kind of like where they are and they're helping those fibers kind of find their space? And it's very possible that we know that already, or we know more about that than we did 5 years ago. But that's the extent of what I know. But it's a really interesting question. And also, as you grow, right? Like a baby has an arm that's like this long. And then an adult has an arm that's significantly longer. And it's the same neuron that has to grow, you know? So basically what the point of what I'm saying is that Schwann cells are very plastic. They can do this thing called dedifferentiation. So going from a myelinating cell to this this repair Schwann cell phenotype. And wouldn't it be great if we could figure out how to make oligodendrocytes do that? Because they don't. And they like require that the environment be in such a way as to support their production of new myelin. There cannot be myelin debris, there cannot be inflammation. The oligodendrocytes, they need a specific situation, a specific environment where the Schwann cells are a little more scrappy, I think, so to speak.
So why is it good for us to know, just to understand the mechanism, or would we use it in some way?
Well, that's the tricky thing. Like in this paper, they're going to be taking out this protein, this F-box W7, and they're going to see like what happens to the myelin, what happens to the Schwann cells. And based on what they found with similar knockout experiments in zebrafish, which is often used as a model for myelination because you can make them clear and you can like just see through them. And it's really easy to make different cell types fluoresce, even in the living animal. But based on those studies, it seemed like if you remove this F-box protein, you increase myelination, which you're like, oh, that'd be great, right? More myelin. That's just what I was talking about. Like, how do we make oligodendrocytes like myelinate more? But more is not always better, And so sometimes you would have things like oligodendrocytes are only supposed to myelinate axons. Like they don't need to be putting their fingers anywhere else. But, mess with a few things. You might get, as they say in here, like enhanced myelination potential. But suddenly the oligodendrocytes are like myelinating dendrites and cell bodies and blood vessels and
myelinating blood vessels really have no bad idea.
I don't know if they do that. It would be a really terrible idea. But I'm not going to go down that rabbit hole.
Okay. That would be really.
But Jonah Chan's group, he's one of the senior authors on this, showed that they could culture oligodendrocytes in a dish that had these carbon nanotubes in them.
I've heard about those.
And that the size and shape, so like the physical properties of those carbon tubes, was enough for the oligodendrocyte to kind of feel it and be like, oh, okay, I can myelinate this. Whereas they would avoid things that had different shapes and properties. And then if you like knock this thing out or overexpress that thing, then the oligodendrocyte would start being like, oh, look at this other shape. I'll wrap around that. And that's definitely maladaptive. So I think that papers like this one, they're really focused on unearthing fundamental processes and fundamental or the mechanisms behind decisions about like what is the cell going to become? How is it going to behave in its environment? But that means that it's quite a few steps back from like, how do we apply this? But if this group and the other groups associated with it, they're collaborators, are chipping away at this, then probably we're getting closer and closer to understanding like the finer points of these phenotypes, like with enhanced myelination potential or like more targeted myelination potential. Understanding that, and then maybe from there we can figure out how to, like, in a targeted way, apply it in vivo.
Okay.
But yeah, so we already talked about how these different trying to think if there was anything else about the Schwann cell oligodendrocyte difference that is worth mentioning. But basically, I mean, these two cell types come from different places in terms of lineage. So I think it's easy to be like, oh well, they're just different.
Yep.
Case
That's what I
closed.
thought
But
going in, is that they somehow come from the same progenitor and they specialize into different things, but they turn out to come from completely different progenitors,
Yeah.
which is kind of weird. Is it like a case of conversion even? Because they express the same protein, like myelin basic protein, which is important for formation of myelin. So yeah, I don't know how to think about that.
I mean,
Maybe it's not so crazy.
they come from different, you know, like when the embryos start, or like the, you know, when life begins, it's all from one cell.
Sure. Yeah.
You know, and you know, and then you have a ball of cells, and then as the thing has a 3D shape within the context of, you know, the uterus, it's different parts of that clump of cells are going to be further or closer to sources of nutrients, and this is where you start to have like things kind of dividing or branching off from each other in terms of lineage. So you're right in that at the very early stages, like when this kind of like layer of cells begins to like fold in on itself and create the neural tube and the neural crest, like at one point they all were the same
Okay.
cells, right? They're and but then yes, there's this patch of cells called the neural crest, which give rise to the Schwann cells. But yeah, I mean, eventually, or like if you go far back enough, they were all the same. So like, I don't know anything about how, like, where that first branching off happens and why it happens, but that I feel like is known because it's pretty easy to study those things with like in situ hybridization in embryos, because you can have as many as you want, you know.
But it seems like what they're trying to look at in this paper is that when oligodendrocytes and Schwann cells, when they start to make myelin, do they express the same programs, so to speak, right? Perhaps.
Yeah, I mean, so what they're asking, oh, I don't know if I emphasized this. I can't remember if I did, but it is going to be important. And they really, the same, they're not doing any, they don't do any studies that focus on oligodendrocytes. They draw a couple, like the parallel that they, or the similarity that they point out is the expression of myelin basic protein, or MBP, which is the like, if you look at all the proteins and what proportions they are, the proteins in the myelin, just like I said, it's mostly lipids from the cell membrane. MBP is the one that's expressed most highly. And that's like the marker that we use for myelin. And it has an important role for kind of like zipping together the different layers of the cell membranes, you know, as they wrap. But they don't do any manipulations of oligodendrocytes in this paper. So I don't know if that
okay
kind of addresses your comment, but I wanted to say that Schwann cells have a kind of like monogamous relationship, at least it's a one-sided, non-mutual monogamous relationship with axons. One Schwann cell will myelinate one axon. Now an axon will have multiple Schwann cells on it myelinating it. But there's that kind of one Schwann cell to one axon ratio that sets it apart from what we see in the central nervous system and with oligodendrocytes, where one oligodendrocyte will myelinate many different axons. And then an axon could be myelinated by multiple different oligodendrocytes. And it's important to understand this ratio because when you remove, as they show, when you remove this F-box protein, that things change. And then I'll also circle back to what Tim was talking about, that some Schwann cells will myelinate an axon and others will kind of just like, you know, sort through the different axons and like gather them together. And it's kind of like, like holding on to them, so to speak. But it's not actually, they're not wrapping their membrane around the axons and like compressing all of the cytoplasm out of it and creating these like, thick layers of myelin. And those bundles of axons that are kind of gathered together by these non-myelinating Schwann cells are called Remac bundles. And they're going to be usually kind of smaller diameter axons. So we have like all these different kinds of fibers, all these different kinds of axons, and some of them are real tiny and others are real big. And so the degree of myelination has to do with that diameter. So oftentimes myelin scientists, when they're kind of assessing what is the impact of this manipulation on myelin, it'll be to look at the ratio between the thickness of the myelin and the diameter, because we're expecting it to be a certain way. And if it shifts, then it indicates that there's something different about how either what's happening on the axon, how it's like saying, I need myelin, or how the myelinating cell is understanding the needs of the axon, so to speak. So let's jump into the paper. Because I think there's not, the only other piece of background that I think is that we have to talk about is just very briefly what this F-box protein is. So it's F-box and WD repeat domain containing 7 protein is part of a system that's involved in tagging proteins that need to be degraded. So the cells are constantly making proteins. Some of them are not made correctly and they need to be taken care of. Others are made in high amounts, but then degraded quickly. There's high turnover. And that would happen in a situation where like the cell might at some point need this protein badly, but we don't need it right now. But we just want to make sure that like it's always there. And then others have to do with like, kind of a, pass go or do not pass go kind of situation. Like a cell can go from it has to be, as a precursor. And then some signal comes into the cell that says, okay, you need to degrade these proteins that are involved in maintaining the cell in a precursor form so that it can then advance to a more mature state and start doing whatever it is it was born to do. So that's what this F-box protein is involved in doing. And putting on the tag that says,
just in case,
you should eat this.
And just in case people knows about this, the tag it puts on is ubiquitin, which is the trash. This is trash tag, right? The label that's
slap it on there. Yeah, it's a very cool system, I have to say, because the location of the ubiquitin tag and the number of tags is also meaningful. So we don't need to go into that, but it is a cool system. And so they chose to look at this protein in the context of Schwann cells because, like I mentioned, there's work in oligodendrocytes that say, this F-box W7 is kind of like pumping the brakes. It pumps the brakes on myelination in oligodendrocytes, and it seems to do this by pumping the brakes on a signaling cascade called mTOR, which does too many things. It's like in the Terry Pratchett books. I don't know if anyone has read those, but it's like, what is that? Like the answer to the universe is 42 or something.
Yep, 42.
I think that's Douglas Adams. That's from Hitchhiker's Guide.
Oh, is it Douglas Adams?
Yeah,
okay,
that's more scientific than Terry Pratchett, which is more wizardry. The answers probably in Terry Pratchett would be like,
I think I read
62.
them at the same time, but so they kind of as a child, so they kind of go like this.
Up.
But the answer to the universe is not 42; it's mTOR, which like it's involved in so much, and it's kind of like it's like a bait and switch. People are like, "Oh my god, mTOR is involved," and then they're like, "Oh, okay, wait, yes, it's involved, but."
I've
it actually doesn't really matter.
Rumors mTOR is how all the tech bros gonna live forever.
Yep. I'm seeing Instagram posts of somebody said, I've been taking mTOR in my biological age.
mTOR inhibitor. mTOR is it's
Yeah,
a
the
protein,
inhibitors, yeah.
and the inhibitor they think is important.
Maybe
is
rapamycin. Yeah, rapamycin
Mhm.
is the inhibitor. So they're taking
Oh my
rapamycin
God.
in their
They have
biological
no
age is
idea what they're
reduced.
doing.
Nope.
No idea what they're doing. But mTOR is kind of has been classically studied in the context of sensing nutrients and energetic resources. So it acts as a regulator of cell growth and accelerates cell growth or slows it down. And so in zebrafish, the removal of this FBOXW7 leads to an overexpression of MBP, that myelin protein, and it increases the number of oligodendrocytes and it increases the thickness of the myelin. So they're like, okay, well, maybe this FBOXW7 does the same thing in Schwann cells. So here's the kind of like 30,000 foot view of this paper. What does this protein do in Schwann cells? Okay, so we've talked about this before. When you want to know what something does, you remove it, and then you quantify things about the cell that you've removed it from. So they use the Cree-Lox system, which we have talked about before, but it's just a way of specifically cleaving something out of the DNA, like in a cell specific and sometimes like condition or like time specific way. So I'm not going to get into it, but they drive the excision of FBOXW7 with a promoter that is specific to Schwann cells, called Desert Hedgehog, cousin of Sonic Hedgehog.
Of course.
No.
And then they quantify things about Schwann cells in the sciatic nerve. And I talked earlier about this, that the sciatic nerve is a mixed nerve, so it has incoming sensory fibers as well as outgoing motor fibers. And they don't talk about this here, but I believe there was a follow-up paper, or maybe it was just related. I'd have to look for it again, that dug into how motor and sensory fibers are myelinated differently. Like, what is how do those decisions get made, and how are the relationships between the Schwann cells and those different kinds of fibers um unique? So it's an interesting question. But anyway, so like, what are the things that they're quantifying in Schwann cells? So they look at Schwann cell number at different stages. So they have um 3 days after birth, 21 days, 42 days, almost 6 months. They have like a variety of times. And then they look at the number of axons in the nerve that are myelinated. They look at the thickness of the myelin. And what did they find? They find that when you knock out FBOXW7 in Schwann cells, there's an early increase in the number of Schwann cells. But that just is kind of like there's like a spurt of production of these cells, but it kind of maxes out. So, like, if you look later, a couple weeks later, the control animals and their the mutants, have the same number of Schwann cells overall. The proportion of axons in the nerve that are myelinated is also higher in general, except for like really small axons, so that just indicates that there's a particular pathway involved in sensing the really small axons and the less small axons, and like deciding who needs to be myelinated. The number of axons does not change. So we're not in some way, by removing this protein from Schwann cells, like totally messing up the number of neurons that are, putting their axons into this nerve. They also found that the myelin was thicker in the absence of FBOXW7, which recapitulates what was seen in zebrafish and oligodendrocytes. And they also showed that the Remac bundles, so that's where a Schwann cell kind of like gathers together some of these axons. I just like think of it as like just somebody just like holding a bunch of pool noodles, that those bundles contain fewer axons. And then like the big finding is that in all of the animals that they looked at, all of the nerves that they looked at, they found instances of Schwann cells myelinating multiple axons, which they do not do. They were doing it in a way that was reminiscent of oligodendrocytes. And further, these multiple myelinators had also kind of collected these smaller diameter axons and were kind of like hanging on to them, like in a kind of weird Remac bundle situation. So now you have hybrid Schwann cells, ones that are like kind of Remac.
Hmm.
They're myelinating, but they're also myelinating multiple axons. So this, just by removing this FBOX W7 protein, we have like completely changed the phenotype of the Schwann cells, which I think is like pretty darn amazing. And they show also that I said that they looked at animals that were like 5.5 months old, and this phenotype persisted. So it's not just some transient developmental stage where like the Schwann cells like can't decide who they're gonna be, and then finally they figure it out. It's not like that. It happens and it stays that way.
Vivian.
Yes.
So I have a question.
Mhm.
So as you mentioned, in normal wild type mice, your Schwann cells can be one of two types. One is it would wrap around a single axons and it will form a big giant myelin sheath. And those are called myelinating Schwann cells, perhaps. And then the other type is it would wrap around gather a bunch of very small diameter, small caliber, as they said, a bundle of small, tiny neurons, and then still wrap around them, but it would not myelinate. It would just kind of grab them together. And these are called the Remac bundles. And then when you knock out this FBOXW protein, cells start to wrap around multiple axons and form myelination. And also sometimes the same cell would both form myelination and also form these Remac things. That was just to summarize what you just said, because that's important. That's like the main finding of this paper. So I'm wondering, in wild-type mice, when a Schwann cell quote-unquote decides to be either a myelinating Schwann cell or a Remac Schwann cell, is FBOXW different in these two types of Schwann cells? And are there gene programs
I don't
that are
know.
different?
Did either of you look that up? I did not look that up.
No, I didn't. I didn't look it up.
No, I don't know.
Are those two different of Schwann? Are those two types of Schwann cells kind of from a gene expression point of view different? Do they cluster differently?
I think they do, but I couldn't tell you what what are the main characteristics at the RNA level that we would use to cluster them. But that's a really interesting question. I think something to look at, because I don't know, and it could I just don't follow the literature or haven't in a long time, but maybe that is something that we know now.
Because I wonder,
it becomes an interesting question. Does that go all the way back to precursor cells?
Before a Schwann cell just quote unquote decides whether it's going to be a Remac Schwann cell or a myelinating Schwann cell, when it makes a decision, does it actually actively makes FBOXW? And then before it does it kind of exists in this state, something like that.
Yeah, I'm not sure. And I think you can even go a step back, because there are precursor cells that give rise to Schwann cells, as well as satellite glia, which live in a kind of more an upstream part of the nerve called the DRG, dorsal root ganglion. And in the DRG, these satellite glia serve the role of astrocytes, but they also give rise to Schwann cells, and they do have a kind of stem cell-like quality in certain cases. So if that's where the Remac and the myelinating Schwann cells are coming from, is it that there's some heterogeneity in the satellite glia pool from the very beginning that says these satellite glia are going to give rise to Remac or myelinating, or is it some biological clock or an environment-based decision?
Is it the scientific finding is that Schwann cells in my pinky toe actually came from progenitor cells that lives in the dorsal root ganglion up in my lumbar spinal cord, and they have to go all the way down there.
They're not born.
Oh, okay.
Well, I don't know. It's possible that they.
Oh, that's a long way to migrate.
Exactly, where? Yeah, it's a long way to migrate. But I think that the mature Schwann cell, whether it's whether it's going to be myelinating or non-myelinating, is I would guess that that's the thing that's doing the migrating. and the traveling, not the satellite glia.
Mhm.
But I'm not sure.
Okay.
Interesting questions.
So Vivian,
Mhm.
in Schwann cells and oligodendrocytes, they both have FBXW,
Mhm. Yes.
and it's on.
I can't tell you the timeline of it being turned on or if it's sustained.
Okay.
I feel like I'm gonna have to do
But
some research.
it must be on
and,
because if
like,
you knock it out, all these weird things happen.
but it could be that, I mean, they're knocking it out. Let's see. I need to look and see it.
Oh, they knock it out
If so,
whenever
this
Desert
system
Hedgehog
is
turns on. So apparently that's every
exactly.
omic
It's
like
not
12,
an
E12, something
inducible.
like that.
Yeah, it's not an induce. I think it's it's early, very very early. It might even be earlier than that, but I'm not sure. But yeah. So the Kree-lock systems can be inducible. So you can be like, okay, at this time we're going to turn the enzyme that does the cleaving of the DNA. At this time we're going to turn it on by administering a drug. But in this animal, the enzyme that does the chopping is constitutively expressed. Like as soon as the desert hedgehog
Hmm.
gene program starts coming online, which is when we get Schwann cells. So these are Schwann cells that have always been without FBOXW7, which is why it's important to understand like what were their numbers when they were born. Is there a difference in the kinetics of their production or how they you know migrate along the nerve? But if you were to remove FBOXW7 later in their development, maybe it wouldn't. Maybe it would or it would not influence the myelination potential, because it is possible that if it's consistently expressed, it's always kind of like the break and being like, no, you will only myelinate one thing. Then yes, removal at any time could lead to weirdness. But if it's only
Just
present
because,
at like the beginning to initiate some sort of developmental program that leads to being one-to-one myelinating or multiple myelinating phenotypes,
yeah.
then yeah.
So the question of whether it's on in both cell types is important because the oligodendrocytes, they myelinate many axons, right? So
Mhm.
that's what you get when you turn off FBX7 in the Schwann cells. So it could be off in OLs,
Yeah.
but they say, yeah, I don't know. I guess we
I think the question of when it goes off could be important
yeah, okay.
in determining that, because like if FBOXW7 is maintained, yeah, I mean, it would be interesting to see if it was maintained even in like mature oligodendrocytes, because you can
Yeah.
have like a protein be present, but then there's some other like parallel signaling cascade that's going to kind of like mask it or silence it. And this is where things get complicated, right? Because you can't actually look at any one single signaling cascade and be like, that's the answer. And in fact, the people, the authors of this paper say this. They're like, they did a little dissection of what what targets of FBX W7 might account for this multiple myelination phenotype. And they were like, this is just one of many targets of the FBOX protein. So it could be any of those. It could be more things.
So it appears to be expressed in both cell types. Previous data, I just looked it up. So it's both in
Does
OL
it say
and,
when though? You see anything? Basically, you'd have to look at like a single cell RNA-seq paper of like, or maybe
yes, there is
yeah.
RNA-seq data confirms that FBXW7 is expressed in SCs at both P3 and P21.
Yeah. And so then it might be interesting. It would be
Well, that's
interesting to see
SC.
the oligodendrocyte, the profile
Yeah,
of
the
expression.
OLs, it says it's expressed, but it doesn't give me a time.
Yeah. So TBD, we can dig in or look into that. Okay, so these interesting phenotypes, the instances of what we're seeing, it counts for about 12% of the Schwann cells. But they make a point of saying, and I like this, that this could be an underestimate, because it's really hard to see inside nerves, you know? it's really hard to see inside myelin sheaths, which is why we have to do something called electron microscopy. And we usually get cross sections of axons. So you'll see, like an axon is a fiber, but if you chop it this way, you get a circle, and then you look at the myelin around it. It's just these are kind of just like they're just physical limitations of investigating these structures. It's kind of like those scrolls that they find in like Egyptian tombs that are like so old that if you tried to unroll them, they would just like, so they have to find interesting ways to kind of like be inside, so it's the same way. But so there's a lot of stuff in the nerves that you'd have to unpack. And so you would have to take very, very many thin sections of the nerve and then kind of like reconstruct them after the fact to like see the whole 3D situation of the nerve. And that is labor intensive, very labor intensive and expensive. And it relies on something called electron microscopy, which is like such a pain in the butt.
I think AI is helping quite a bit.
To prepare. Oh, yeah. And like, I remember a handful of years ago, I went to SFN, Society for Neuroscience conference, and there was this like machine that was like, was the size of like a large refrigerator. It was a machine that allowed you to take serial sections prepared. So like 1, 2, 3, 4, like on a deli slicer, serial sections, EM sections.
Wait, I can cite the whole name since they used it in this paper. It's called serial block phase scanning electron microscopy.
Mm-hmm. Yeah. But only at the time, like only like 2 labs had the capability to do
data,
this. And
like
it's
even just the data acquired is going to be insane.
The data,
let alone trying to decide, is it a nucleus, is it cytoplasm, what kind of cell type?
Yeah. And that's also a good point. So like, not only do you need somebody who knows how to prepare the sample for EM, electron microscopy, but then you also need to have somebody who either can do it themselves, which they won't, who can train the computer to recognize different cell types based on like, what does their nucleus look like? What does the density of their cytoplasm or the placement of the endoplasmic reticulum look like? And those are the things that we use typically. It's just like physical structures to identify different cell types. Like the astrocytes, for example, have like large nuclei and kind of lace-like chromatin versus microglia, which have like much smaller, denser nuclei. It's not easy. So basically their point is that they are seeing this in about 12% of the, or in all of the, it accounts for like 12% of the myelinating events that they see. But it's probably an underestimate just because of what they're up against in terms of looking at and analyzing their samples. So they try to get around this as best they can by doing a co-culture of Schwann cells and the neurons the dorsal root ganglion neurons that they would be myelinating in vivo. And they observed the presence of mature Schwann cells, so the Schwann cells that are making MBP. And they know that they're Schwann cells because, well, they express a protein called CROX20, I think. CROX2?
CROX20, I believe.
I want to say 20.
Let me double check.
Yeah, I thought it was 20. Yeah, great. But it's a lineage marker for Schwann cells, and it's not expressed by oligodendrocytes. Because there's always this. Well, I'll just finish up with the culture results, which is that they found the presence of these mature Schwann cells, MBP positive Schwann cells, that had more than 3 MBP positive processes. So those are the ostensibly myelin producing branches of the Schwann cells membrane.
In the knockout mice.
And
in the knockout Schwann cells.
Right. Knockout cultures. Then they also ruled out the possibility that the cells that were doing the multiple axon myelination were oligodendrocytes that had somehow like breached the barrier between the CNS and the PNS, and that they had infiltrated the nerves. And this has been like, this is definitely a place where like myelin researchers like have fought about this. They're like, oh, well, it's the Schwann cells that are coming in, or like, oh, they're not microglia. It's just macrophages that have somehow like made it in through this barrier and have infiltrated the CNS. And even using lineage markers can sometimes be tricky. And like reporter animals based on the presence of these lineage markers. But they showed, but so it's always good to have like another way to show that these are not that, to rule out the possibility that they're oligodendrocytes and not Schwann cells. So they use CROX20 to say like this is a lineage marker for Schwann cells. But also if you look at the samples through EM, they can see the presence of what's called a basal lamina, which is a kind of specialized mixture of matrix proteins or like connective tissue kind of proteins that are found on the outside of the Schwann cells that wrap the axons. And Schwann cells are known to make basal lamina, but oligodendrocytes are known not to make basal lamina. So they had this kind of they had this like sort of like physical this physical structure that helped them say, yeah, these are Schwann cells, in addition to having a lineage marker that they could look for through immunofluorescence. And so this, the next thing that they're going to do is look at the kind of functional outcomes of the increased myelination potential, because we were like, oh, you boost myelin production, isn't that great? Like more myelin, yay. But not necessarily, because by manipulating this FBOX protein, you have dramatically changed the situation in the nerve and the cells that are in there. So like what outcome does that have on behavior? So they took these roughly 6-month-old animals, and they did a couple of tests. And the take-home message is that the mutants, they had this vertical pole, and they put the mice on the top, and then they see how long it takes for the mouse to climb down the vertical pole. And the mutants took longer to crawl down the pole than the controls, suggesting that they have a deficit in complex motor behaviors. They also did a gait analysis. So they have the animals like walking on this little catwalk, and you can look from underneath and see like where you can capture all sorts of information about their gait. And basically the mutants walk funny.
It's a bit of a shame
So
they didn't do like nerve conduction study to see if the speed has
that's
changed.
yes, that would have been interesting. Yeah. Yeah, that would have been cool, because then they also, so the primary like dysfunction that they found was motor. And then there was this trend towards hypersensitivity to cold. But it didn't reach statistical significance. But yeah, to your point, I think it would have been cool if they had like been able to get in there and do a couple of different kinds of stimuli, and then see how activity in the nerve changed. So the next thing to do is to look at mechanism. We knocked the thing out. Stuff happened at the level of the tissue and the cells and the cell morphology. It had an impact on behavior. So let's go back to the cells and to this protein and say like, well, what pathway is it part of? So as we mentioned before, there's already this connection between FBOXW7 and mTOR. So if you knock out FBOXW7, you get more mTOR signaling. And similarly, if you get mTOR protein, they found was elevated in the Schwann cells of the FBOXW7 mutants, as were the downstream targets of mTOR. So FBOX goes away, mTOR goes up, mTOR targets also go up. So this begs the question, if you've lost FBOXW7 and it leads to increased mTOR signaling, if you now take away mTOR, can you take this mutant phenotype and bring it back to kind of what the control animals look like? So they now are going to create double knockouts.
Can you fix a
Still
broken
under-
system by breaking something else?
Sometimes
Yeah.
you can.
Yeah. It's like
But that's how you test.
you know,
That's
like
how you test
sometimes they would
mechanism.
call,
It's like, whatever is broken, does it depend on something else? Yeah.
yeah. And what's really cool about this is it's like yes and no. Because I always like I don't know why this speaks to me, but I always like that it's cool for me to think about how the different stages of a cell's life arc, we tend to think of them as like all being connected. Like you go from being like you just transition from being a precursor cell into a mature cell that can do all the things. Yes, if you turn off the things that that prevent the cell from progressing to the next stage, you can drive it to become a mature version of itself. But that does not mean that they can pass go with whatever else they should be doing. So like you can have a mature oligodendrocyte that has not yet gotten the signal that it needs to produce myelin. So these different things these different stages of the life arc that we kind of perceive as being all connected are actually like individually regulated stages. You can, so I think that's kind of cool. And what they show here is that you can pull apart this phenotype that they see. So they do this, they do double mutants where they've knocked out, they're not quite the same as the original single mutant, because if you do full knockouts of both mTOR, like we have 2 alleles of every gene, and if you knock both alleles of mTOR out and both alleles of FBOXW7 out, they said they had like 1 pup out of 126 pups that survived. So that's clearly not compatible with life. So, but if you take out just one allele of FBOXW7, you still get the phenotype. So like Schwann cells, the interesting Schwann cell phenotype, but the animals survive in the complete absence of mTOR. So they find that some of the things that they quantified earlier in their single mutants do return more or less to control levels. So that early increase in Schwann cell number, that goes away. The percentage of the increased percentage of myelinated axons also goes away. And the kind of like hypermyelination, the increased thickness of myelination also diminishes. But the Remac bundle, and so like on the flip side, the Remac bundles in the double mutants actually it's like suddenly they've collected even more axons, so you've got these like large Remac bundles. But they found that so mTOR does appear to be part of those changes, those phenotypes. But loss of mTOR did not push the Schwann cells back to their like monogamous state. They still found instances of Schwann cells myelinating multiple axons. So the loss of mTOR was not sufficient to, as I say, suppress the enhanced myelinating potential of the Schwann cells. Okay, so if it's not mTOR, then what is it? And so they went back to, as we just did earlier, they go and mine some RNA-seq databases and to look for targets of FBOXW7, because if FBOXW7 is not doing its job, then other things are going to go up, things that it usually would be saying, hey, you should get rid of this. And they found a couple of different targets, but one that caught their attention was this protein called c-jun. And the reason that they were like, oh, we should look into c-jun is because c-jun is part of the dedifferentiation program that follows injury. And because we have to take myelinating Schwann cells and make them into repair Schwann cells that have a multipolar morphology. And the kind of, these hybrid Schwann cells that they found in their FBOXW7 mutants are multipolar. They have multiple arms that are going out and contacting axons and starting and initiating wrapping. So they used immunofluorescence, which is like you tag your protein of interest using an antibody that recognizes your your target, c-jun, and you've connected that antibody to a fluorescent molecule, and then you can look at it under a microscope. We've talked about that like a zillion times, I'm sure. So they showed that c-jun levels in the absence of FBOXW7 were significantly higher, like triple the levels in these Schwann cells than they are in control animals. So that's pretty compelling. So at this point, it stops there, because if we were gonna dig deeper into mechanism, then it would be like, okay, well, let's manipulate c-jun, or let's figure out what genes is c-jun acting on. This, you can always keep following the trail of breadcrumbs. But we've reached the end of the paper, so like the bottom line is that the model that they develop in this work is that FBOXW7 is pushing this one-to-one myelinating characteristic that we've seen in myelinating Schwann cells by suppressing c-jun signaling. So then if c-jun is allowed to increase, it kind of like pushes the Schwann cells to a more like a, plastic kind of repair like multipolar like phenotype, and that this allows the Schwann cells to now be myelinating multiple things. They didn't look. I'd have to maybe dig a little bit more deeply and like look at the images closely, but I don't believe that they mentioned anything about like the integrity of the myelin being different.
What is myelin integrity?
The degree to which the different layers of the myelin are zipped tight.
Okay.
And if if you have an axon and the myelin is wrapped around it, it has special there's a specific like expression pattern of molecule or proteins at this end and at this end. And if that's messed up somehow, then you'll have like the ends of the myelin of the membranes
Fraying out.
lifting off of the axon. Okay. You might have like bubbles in the myelin where for whatever reason it's not been able to like push out all the cytoplasm.
So you mentioned earlier that during myelin formation, like all the cytoplasm juices get squeezed out, so that it's just like a cable. And myelin basic protein is responsible for like mechanically squeezing everything out.
Yes,
okay.
It and one other protein called PLP,
okay.
proteolipid protein, and I can't quite remember, but it's like, so you've got. These are all like notoriously difficult to explain. If you see it in a picture, I think it helps. But like, if you have a cell and if you zoom in on its membrane, it's got two layers of lipid molecules. Okay. The lipid, bilayer. So I can't remember if this is PLP or MBP, but some protein has to be involved in like smushing those two lipid layers together. And then as the cell wraps, you've got another lipid bilayer that has to be smushed. And then you got to smush those ones together. And PLP and MBP do the smushing, but one of them is involved in smushing the bilayers together, and the other is involved in smushing the smushed bilayers together.
I see.
So I looked up, and it says that c-jun is a negative regulator of myelination in Schwann cells. Mm-hmm. So,
how do you get multiple? That's why I bring up the integrity thing.
Yeah, but I don't know why it goes up when you knock out the box gene, because going up should even more negatively regulate myelin. So it doesn't make sense.
No, I mean, it doesn't, but that's why they say
it depends.
At the end that there's a lot more.
But like, for example,
happening.
myelin thickness seems to be governed by mTOR, which is separate to c-jun.
Yeah.
And c-jun might be more important for you to be like multipolar in terms of your your Schwann cell phenotype.
Yeah, I think it's kind of like, it's like a mobile where, the things on the mobile will like hang at a certain angle unless you add something else, and then it shifts it to a slightly different position. And that's kind of how I see if you're only considering like, something very simple and you're only considering 2 signaling cascades. Yes, this is how it's going to look. But as soon as you like pay attention to the fact that there's a third signaling cascade that's coming in and like modifying the outcome of these 2 signaling cascades combined, then it just gets increasingly complicated. So, but I think like it'd be interesting to see if a follow-up paper got at your question to be like, okay, well,
Yeah.
if c-jun is so high that it's creating this multipolar phenotype that is similar to a repair Schwann cell, how is it that they're still creating myelin? And I guess the question might be like, is it actually like bona fide myelin? And I mean, I think that I don't see anything here. They haven't said anything that suggests that the myelin is not stable or that it's organized improperly. But they also, they deal with the obstacle of how to analyze, how to analyze these samples because of the physical constraints.
I have a couple of questions because we're done. We've kind of wrapped up the paper, right? So we can start.
We've wrapped up the paper.
Sorry, pun intended. So they mentioned that when you knock out this F-box WD7 protein, which is the whole point of the paper, then the Schwann cells assume this multipolar configuration that's similar to like an injury kind of phenotype. So the obvious question is, when you get injured and the Schwann cells do assume this multipolar phenotype, is the F-box protein go down? And I don't know whether, I mean, they didn't address it here, but I'm just wondering, because if it doesn't go down, then how come c-jun can be elevated despite this F-box being a negative regulator?
Well, because everything's got a brake and a gas pedal, and they're not necessarily in the same.
Okay.
And this is not a great analogy, right? But like you can have the brake, this one particular brake on c-jun, the F-box W7, like it can be there. But if you have a stronger gas pedal coming from a different place, that might be enough. But I think it is a really interesting question. Like, if c-jun goes up, how is it that they're still? How are these cells still myelinating?
Yeah, and how does it even go up in the first place? But then the second question is, at the beginning we talked about this F-box W7. It's hard to remember. WD7. That's a new lubricant. How this F-box protein is ubiquitous. It kind of tells ubiquitin ligase, kind of the trash tag label where to go. But then we didn't really talk about how ubiquitin plays a role for the rest of the paper. And in fact, when you remove this F-box protein, it actually led to, because the most obvious and most naive way that I would think about this is that, F-box negatively regulate mTOR because it causes mTOR to get degraded via ubiquitination. But then it actually causes the transcript of mTOR to go up. So it's even like one step upstream of mTOR. It's already going up. So it really suggests that this F-box
inhibiting
it is some systemic thing going on where it's maybe degrading like transcription factors or like a broad set of programs. And I'm just curious as to like how does the ubiquitination figure into this? But I don't guess they didn't.
Yeah, I don't know. They didn't go into it, but but like, so that's the problem. Like c-jun is one of these. There's some like, what is it called? Oh my God, I'm totally blanking. So like
immediate early gene. It's like
yes, immediate early genes.
It's like one of the AP1.
Yeah, so like there's it's mTOR. I think c-jun is one of them, or Jun is, yeah, and it's like once they go on, that there's just a bunch of other stuff that's being unfurled. And we like in a paper like this, I mean, they're reporting what they're seeing and that they did this thing. And so there's got to be some connection. But we also all know that a lot of other things have just been triggered. And like I was saying, in different ways. Like you have both like simultaneous like pressure to not do it and a drive to do it. And it's just the balance of that balance that's going to decide what the phenotype is, what the outcome is. Because nothing is like a step function, like it's off and then it's on. It's all about like, if you're mixing a cocktail, like, are you heavy on the this and light on the that? And, this version's going to lead to more inebriation than this version, So
I looked up this lab and they haven't done anything since then.
On the
See,
F-box.
okay, this is what...
Yeah, on the myelination in the F-box with c-jun and nothing. No.
It's, what have they been working on? The astrocytes.
Hold on. I'll look. So here, well, there's nothing after 2019 you want to know, right? Because that's when this paper was published. There's this, well, I put most recent. Let's put publication date. So in 2026, pre-myelinating oligodendrocyte ADG-RG1 is required for axon ensheathment.
Mm-hmm.
Then voltage-gated channels regulate myelination. There's another cannabinoid receptor leads to aberrant myelination.
Mm-hmm.
A lot of stuff.
Yeah, see, so like there was this guy, Vittorio Gallo, who's I don't know where he is now, but he was at George Washington. And what I can't even remember because this was like in 2018, 2019. But I was like helping to write some kind of like homage to him. I can't even remember why, but I went and looked at all of his publications, and I was just like, I thought it was so great that he had just like over the decades, like just chipped away at the story. He's studying potassium channels, and it was like, we make this step forward. We make this step forward. We make, and he had just like worked really hard and diligently to like illuminate this channel, no pun intended, but like this line of inquiry. And I just don't see that enough anymore. Because it's like,
Yeah.
Oh my gosh, don't even get me started on this whole funding and
like whatever your research takes you just takes you there. It's hard to.
Here's another paper.
Yeah, but you gotta exert control on that. You can't just be wandering around and be like, oh look, a pretty butterfly, oh a pretty flower.
2025. Another so FBXW7, which we've been talking about, regulates MYRF levels to control myelin capacity.
Myelin regulatory factor.
So it's
which is a major transcription factor.
So I guess it's regulating it by degradation, ubiquitin pathway, right?
Mm-hmm. Mm-hmm.
So that's the follow-up. Yeah, 2025.
Yeah, I see that. Homeostasis. Interesting.
Okay, that's
all right, folks. That's
TWiN
a wrap.
70. You can find the show notes at microbe.tv/TWiN, and you can send us questions or comments to TWiN at microbe.tv, or you can leave them on the YouTube channel. You can leave them below. We'll look at them.
Mm-hmm. Yeah, I've been trying to do that.
If you like these programs, we'd love your support. Microbe.tv/contribute. Vivian Morrison is in New Orleans. Thank you, Vivian.
Yeah, thanks, guys. It was fun to return to something a little more less mind-bending.
I don't know. I thought fibromyalgia was cool.
I mean, yeah, sure.
Yeah, sure. Tim Chung is
no,
at
it's
NYU.
Important.
Yes, it's all good. Tim Chung is at New York University. Thank you, Tim.
Thank you. Thanks, Vivian. Yeah, it's good to learn about the neglected, at least in my case, neglected glial cells, because I rarely think about them.
Yeah, and I think also just like some just good old-fashioned, simple science. Simple techniques.
I'm Vincent Racaniello. You can find me here at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.